All right, here we go. So this topic is on translation. So this is the second step in that central dogma of biology with the DNA makes mRNA makes proteins. This is the mRNA to protein part. And translation by definition is the synthesis of a polypeptide using information from the mRNA. So taking a step backwards with transcription, we have the mRNA molecule that was made by the template strand of the DNA, and now this is taking that and translating it into a polypeptide, which will then become a protein. It occurs at the ribosome. It's the site of protein synthesis. Nucleotide sequence becomes an amino acid sequence. You can see as far as like structure of the tRNA goes, you have the amino acid up here, so the amino acid sequence is up top. You have the anticodon down over here, and it's a key player in translating mRNA into that amino acid sequence. So, yep, as far as I was mentioning earlier that it has an anticodon region that is complementary and anti-parallel to the mRNA. So you can see the 3' and the 5'. But more importantly here, you have the uracil that matches with the adenine, the guanine matches with the cytosine, and adenine matches with the uracil here. So it has an anticodon region, which is this region down here. This region being the codon region. So codon, anticodon. All right, carries the amino acid up top here, so that you can imagine that's like the head of the little stick figure guy. And the mRNA codon is down over here, as mentioned. Don't worry about this. The enzyme aminoacetyl tRNA synthase is responsible for attaching amino acids. Don't worry about any of that right there. That's yeah, don't worry about that. All right, cool. All you need to know in this slide is really the anticodon region down over here, oops, on the tRNA, the codon region over here, the amino acid sequence up there. That's the big important stuff. So let's talk ribosomes. So translation occurs at the ribosome that is in the cytoplasm of the cell. There's two subunits to it. You've got a small subunit down over here. You'll notice it's smaller. And you'll have the large subunit up over here. You'll notice it's larger. And what happens is the mRNA molecule kind of gets sandwiched in between these two, and your tRNA molecules kind of hang out inside these sites in the ribosomes themselves. Different sizes, of course, for prokaryotes and eukaryotes to, different organisms. Prokaryotes, even the small subunits are even smaller, and so on and so forth. But all you need to know here is that different size subunits, two subunits in total, one small, one large, and that is the site of translation. Pause here to take notes. Sorry, I forgot to say that for the last one. Cool. So APE are the sites of inside the ribosomes. APE. A stands for the amino acid site. So this is the incoming site. It holds the next tRNA carrying an amino acid. So we have an amino acid here, we have our tRNA here. It's going to go into the A site. All right. The P site over here holds we call it the P site because it's the growing polypeptide chain. It holds the tRNA that is carrying that chain. And then we have the E site, which is the exit site. And what's going to happen here is tRNA is going to come here. It's going to be the amino acid up top. This is going to bind. and create a chain. This whole thing moves over and then out to go pick up, the new one. Cool. Good stuff. So quick check. Pause here if you need to take notes. Quick check. How does tRNA interact with mRNA? How do they hang out with each other? What are their interactions? Through their anticodon. So remember, it matches up A, this would be the codon, C, A, and then the anticodon, U, G, U, right? And they would kind of interact with each other on that level. Boom. What codes for the amino acid? The mRNA codon. What are the 3 sites on the large ribosomal subunit? Let's be honest, you guys just wait till the end and fast forward and copy the answers down. It's the APE site. I'm going to start removing the answers from here. Awesome. A stands for amino acid. It's the incoming amino acid. P stands for polypeptide. That's where the growing polypeptide chain is. And E stands for exit. It's where the whole thing exits out. It occurs in 3 stages. They may sound familiar. They're the same names that we use for transcription. They are initiation, elongation, and termination. So how initiation starts? I'm sorry, go back here if you need to take notes. Initiation, elongation, termination. All right, step by step. Step 1. It begins with a start codon. Just a simple AUG. We see it down here in the mRNA. It's a codon, and it starts the whole thing. First, the small subunit will come in, then the large subunit will come in, and the very first amino acid, methionine, comes in. It all starts with the start codon. tRNA carries methionine. Next, the large subunit binds. It's like step by step. It all starts with AUG on the mRNA molecule. Small one comes in, methionine comes in, large subunit comes in. They all make one big happy sandwich of a family. Note, the first tRNA carrying methionine will go to the P site. The P site. Because this is the one holding the polypeptide. That is the one polypeptide to start. Everything else from here on out will go to the A site. But you can see when this whole thing comes down, this would be the P site. Next codon in here would be the A, and it moves that way. Great. Pause here to take notes. Step 2, elongation, making it longer. So elongation starts when the next tRNA comes into the A site. So you can see here we have our methionine, we have our AUG start codon. Now we have the next tRNA coming into the A site. RNA is moved through the ribosome and its codons are read just one at a time. So AUG starts and it just sort of moves down in this direction. mRNA codon codes for each specific amino acid. And you don't have to memorize any of it. So codon charts will be given to you, but it's essentially 1, 2, 3, 1, 2, 3, 1, 2, 3. And you can see the anticodons on the other side, even down here. Great. Good stuff. So this is one of the sort of supporting pieces of evidence that suggests common ancestry, that we've all kind of evolved from the same thing, is the fact that we use the same genetic code It's just sort of one other piece of evidence in evolution. Because remember, this is true for all living things. Me, you, my cats, the bacteria in the lab, all of it. Pretty cool. Pause here if you need to take notes. Oop, pause here. So elongation occurs in steps. So there's the codon recognition that goes into the A site. So you got the start codon here, codon recognition. So we're making sure that the A and the U and the A and the U and the G and the C. Then there's a peptide bond that forms. Remember, this is a polypeptide. They form peptide bonds. Actually, not this one. This one's going to go here, goes at the bottom, whatever. All right, so peptide bond is formed and it transfers that polypeptide into the A site, to the A site tRNA, right? So you can see here's where we started. This one then moves into the E site. E site leaves. And you can see the P goes into the A goes into the P. See? So it sort of hops down one over the other. And then the translocation. So the tRNA in the A site moves down to the P. That's sort of what I was just saying. tRNA in the P site goes to the E site. A site is open for the next tRNA, and the whole process starts over again, right? So anticodon, the appropriate anticodon from the next tRNA goes into the A site. Bond is formed. Everything shifts down. And then we start all over again. Pause here to take notes. And finally, we are at termination. So termination occurs when a stop codon in the mRNA reaches the A site. So we started with the start codon, we're going to stop with the stop codon. They don't code for amino acids. They signal for a release factor. So something will happen to cause the release of the mRNA molecule. Hydrolyzes the bond that holds the peptides to the P site. So it's a hydrolysis reaction. Peptides release, and all translational units disassemble. So once we hit that stop codon, essentially the bond that is in the P site that holds that peptide in there, that goes away. The polypeptide itself, so the chain of amino acids, releases, and the ribosome disassembles with all other sort of units. All other all other translational factors. Boom. Stop here if you need to take notes. So take a few minutes, pause here, go back, get a pet or a parent. I used to love going to my parents and being like, let me explain to you transcription and translation. And they were like, what? They're like, you're crazy. Get a stuffed animal and explain the 3 stages. Quick review. Before we move on to protein folding, take a minute. Ooh, do you remember this? Primary, secondary, those side chain interactions, tertiary and quaternary protein structures. Yeah, you think unit 1 and 2 going away? Never will. Primary is the chain of amino acids. Secondary, it folds and coils due to hydrogen bonds. So secondary hydrogen bonds. Tertiary is the side chains. And quaternary is 2 or more polypeptides interacting. So let's talk protein folding. Sorry, pause here if you gotta you gotta jot this down. Let's talk protein folding. So as translation's taking place, the growing polypeptide begins to coil and fold. These things happen at the same time. The genes themselves determine that primary structure, DNA to mRNA to protein. So the genes essentially determine, after being transcribed and translated, the primary structure of the polypeptide. And the primary structure, as we know, determines that final shape because it's the order in which those amino acids are put together. Some polypeptides require chaperone proteins. They're exactly as they sound. They help fold correctly and would modify the proteins before it could be functional in the cell itself. So it sort of helps it along. Take a second, pause if you got to take notes. Cool. I'm not going to play this video because I was told that the sound doesn't work, but this will be up on Classrooms, this video, so feel free to click on it. You probably are also going to watch it in class. All right, now let's take a minute and put everything that we just learned aside, because we're going to learn about something that breaks all the rules. Retroviruses. So as far as biology goes, these are probably the most fascinating little things on the planet. So HIV is a retrovirus, and it's the exception to the standard flow of genetic information. Essentially, the information in a retrovirus flows from RNA to DNA. So you can see that up here, so here's a little HIV virus, and it fuses to the host cell. It essentially dumps its RNA into our cell and inserts itself into our own DNA. Then our own DNA and our own healthy, well, not so much healthy anymore, in our cells produce that protein that is then released to create more viral particles. So this is why HIV and other forms of retroviruses are so deadly. They use an enzyme called reverse transcriptase. So it essentially is able to take its viral DNA and insert it into the host DNA, the host being us. It's pretty crazy stuff. We are using CRISPR technology now in some other forms of gene editing, and have seen a lot of promise in curing retroviruses like this in the patient. So very interesting. Definitely keep your ears and eyes open for studies that are coming up regarding retroviruses. This is also a way people are thinking about curing cancer by taking genetically engineered retroviruses to be able to inject new DNA into the cells in order to cut out like the malfunctioning P53 gene, that sort of thing. So very fascinating stuff. Definitely keep an eye out. So here we go. Practice FRQ. This is the one I'm going to be looking for, so please take a minute and do it. All living organisms need a transfer of information to allow for genes to direct the synthesis of proteins. Explain how genes are transcribed and translated to produce a functional protein. Functional protein. So please also talk about protein folding. Okay? Do that. Pause if you need to. Great. I'll be checking for your answer. Practice FRQ number 2. Compare and contrast translation in prokaryotes and eukaryotes. Ooh, that's a great question. Can't wait to see your answers. Bye, guys. I'll see you tomorrow.